Hi students, this video is for FMGE aspirants. So here is a platform for you to revise your PYQs which are high yield for your exam clearance. Okay, so I'll make a series of videos on the PYQs. All right, so let's see the first question. All enzymes are not proteins. This statement is justified by, okay, so we have four options. Of these four options, which option do you think is the justification for the statement all enzymes are not proteins? That is what you have to choose. You should not choose what is the right, which is a true statement. Okay, so true statement is not what they have asked. They have asked the justification for the above statement. Now let's see the options one by one. All enzymes do not follow Michaelis-Menten hypothesis. Do you think it's true? Yes, it is true because you have this, because you have something called as the, so you have something called as the allosteric enzymes that do not follow Michaelis-Menten hypothesis. Yes, so it shows a sigmoidal curve, right? So all enzymes do not follow Michaelis-Menten hypothesis. But this statement does not justify all enzymes are not proteins. Okay. And next option is RNAs act as ribozymes. Yes, RNAs have catalytic activity. They are called as ribozymes. So what are they? 28S rRNA, yes, or snRNAs, peptidyl transferase. So these are RNAs that can act as that have catalytic activity. They are called as ribozymes. So do you think this option can justifies the all enzymes are not protein statement? Yes, of course. Okay. So, but still we have to see the other options. Antibodies take part in the catalysis of many reactions. Yes, do you think that antibodies take part? Yes, there is something called as the abzymes. Okay, abzymes are nothing but antibodies that have catalytic activity. Okay, and metals are involved in attachment to enzymes. You have something called as the metalloenzyme. Yes, metalloenzyme. For example, tyrosinase. The enzyme tyrosinase has copper in it. The enzyme xanthine oxidase has molybdenum in it. The enzyme carbonic anhydrase has zinc in it. Okay, so these are metalloenzymes. Whereas you also have metal ion activated enzymes, or calcium dependent enzymes, or calcium dependent kinases, or sodium dependent enzymes. So these are all metal ion activated enzymes. So the answer is RNAs act as ribozymes. That only justifies the statement all enzymes are not proteins. And the next question is, in oxidative phosphorylation, ATP production and respiratory chain are linked by. So in oxidative phosphorylation, oxidation plus phosphorylation is oxidative phosphorylation. So in electron transport chain that takes place in the, where is the electron transport chain located? It is on the inner side of the inner mitochondrial membrane. So we have four complexes. Yes, complex 1, 2, 3, and 4. So complex 1 is NADH dehydrogenase. Complex 2 is FAD dehydrogenase. Complex 3 is cytochrome reductase. Complex 4 is cytochrome oxidase. Okay, and the electrons from NADH2, the electrons from NADH and FADH2 pass through the electron transport chain. Okay, so flow through these complexes. So NADH, the electrons move from complex 1 and a mobile carrier coenzyme Q to complex 3 and another mobile carrier cytochrome C, and then to complex 4, and then finally to oxygen. Similarly, electrons from FADH2 from complex 2 to coenzyme Q to complex 3 to cytochrome C to complex 4 to oxygen. So the electron flow will be in this manner. That is why it is called electron transport chain. So the two mobile carriers will be coenzyme Q and cytochrome C, and the final acceptor is the final electron acceptor is the oxygen. Okay. Now, as the electrons move from one complex to the other, some amount of energy is liberated. This energy liberated is used in pumping hydrogen ions. Yes, used in pumping hydrogen ions from the matrix to the mitochondria inter membrane space. okay, to the intermembrane mitochondria, space in the mitochondria. So here 4 hydrogen gets pumped out from the matrix to the intermembrane space through complex 1, and from complex 3, 4 hydrogen ions, and from complex 4 to oxygen when the electron flows, 2 hydrogen ions. Okay, so the energy which is liberated when the electron flows from one complex to the other is used up in pumping H plus ions from inside the matrix to the intermembrane space. Now, there is so much of protons in the intermembrane space now, is it not? We have another one complex which is called as the complex 5 or the ATP synthase complex. Complex 5 or the ATP synthase complex. So this now what happens, there is a proton gradient which is formed between the intermembrane space and the matrix, is it not? So now if we are going to dissipate the proton gradient via ATP synthase complex, then some amount of energy will be liberated. That energy is used up in the synthesis of ATP from ADP. Okay, ADP and inorganic phosphate will form ATP. So what is giving the energy to form ATP? The proton gradient. So as you dissipate the proton gradient, which means that all of the H plus ions from the intermembrane space will now be pumped into the matrix via ATP synthase complex. So now what happens? The proton gradient get dissipated. Thus, what happens? The energy is liberated, that is used up in the conversion of ADP with inorganic phosphate to ATP. Okay, so that this is what is called as the chemical energy. Yes, that is chemi-osmosis. Osmosis is what is happening here, is it not? The H plus ion which has been accumulated in the intermembrane space is driven into the matrix via ATP synthase complex. So this is what is chemi-osmosis. This is what is chemi-osmosis. So this in oxidative phosphorylation, the ATP production and the respiratory chain is nothing but the electron transport chain with the final acceptor as oxygen. That is why it's called respiratory chain. That is linked by a chemi-osmotic method. Yes, it is linked by a chemi-osmotic method. So the answer is chemi-osmotic method. Next, phenylalanine is a precursor of all of the following except. So we all know phenylalanine gives tyrosine, is it not? Phenylalanine hydroxylase. So phenylalanine gives tyrosine. So one of the precursor, for tyrosine, phenylalanine is the precursor. So what are the special products of tyrosine? Special products of tyrosine will be thyroxine. Yes. And then catecholamines. So what are catecholamines? Epinephrine, norepinephrine, and dopamine. Okay. And then melanin. Melanin is the skin pigment. So catecholamines, melanin, and thyroxine are special products from tyrosine. And tyrosine is formed from phenylalanine. So phenylalanine is the precursor of all these four, except what? Melatonin. Melatonin is formed from tryptophan. Yes, melatonin is formed from tryptophan. The answer exception is melatonin. ApoB48 and ApoB100 are expressed as two different apoproteins because of which mechanism? This is one of the regulation of gene expression, which is RNA editing. So what is RNA editing? Here we have the ApoB gene. Okay, so here we have the ApoB gene, which is common for both ApoB48 and ApoB100. Now, as the ApoB gene gets transcribed, it forms the mRNA. Yes, it forms the mRNA. Now, this mRNA, in the intestine, what happens? In mRNA, in the intestine, as it undergoes translation, so in the intestine, being in intestine, what happens? The translation of ApoB gene stops at the 48th position. Okay, so because 48th position is stop codon occupies, so the translation of the ApoB gene stops at the 48th position. So making it the ApoB48 protein. Yes, whereas this is incorporated in chylomicrons. So chylomicrons are those which have this ApoB48 and it's synthesized in the intestine. Whereas ApoB100, when the mRNA of the ApoB gene is fully translated, it forms ApoB100, which is incorporated in lipoproteins such as LDL, VLDL, right? So this is what is called as RNA editing. So the answer is RNA editing. So this question, in a well-fed state, acetyl-CoA obtained from diet is least used in the synthesis of, okay, least used. So first you should understand about the fate of acetyl-CoA. So acetyl-CoA is formed from what? Glucose. From the diet, glucose gives pyruvate, yes, pyruvate, and pyruvate gets converted to acetyl-CoA. So in the well-fed state, there is more of acetyl-CoA, and this acetyl-CoA can enter into TCA cycle, and then to electron transport chain, then it can give us ATP, is it not? And similarly, in a starving state, what can happen? It can form ketone bodies, okay? It can form ketone bodies. As well as in the well-fed state, it can also form fatty acid synthesis, cholesterol synthesis. Yes, the starting material is acetyl-CoA for fatty acid synthesis and cholesterol synthesis. So in these options, palmitoyl-CoA is a fatty acyl-CoA. So acetyl-CoA is used in the fatty acid synthesis. So in a well-fed state, the acetyl-CoA, excess acetyl-CoA can enter into fatty acid synthesis. Citrate and oxaloacetate are intermediates of TCA cycle. So of course, acetyl-CoA can enter into TCA cycle, but it is least used in the synthesis of acetoacetate, which is a ketone body. Yes, so only during starvation state, ketone bodies synthesis takes place inside our body. So in well-fed state, it's not starving state, is it not? So in well-fed state, acetyl-CoA obtained from diet is least used in the synthesis of acetoacetate, which is a ketone body. Thank you all. Meet you with another set of MCQs. Okay, thank you.